So in this part of the video, we will talk about the regulation of the secondary metabolism or secondary metabolic pathway. Regulation of the secondary metabolic pathway is very important because again, most of the products of the secondary metabolic pathways have special uses. So unless the cell or the organism is to provide or to expend energy for this pathway, they really wouldn't do it otherwise. So, one regulation is carbon catabolite repression. So, basically, glucose suppression of antibiotics. This is seen usually in the production of penicillin and chloramphenicol. Um, Basically, long story short, the presence of glucose prevents the production of antibiotics. That is also reflected because most of the time that we see the secondary metabolism is during the stationary phase. And what is in the stationary phase? In the stationary phase, you have a lot of organisms, but very, very low amount of nutrients. So because of the low amount of nutrients, the secondary metabolism kicks in, and that's actually the signal for the secondary metabolism to start. That is why in the presence of glucose, the secondary metabolite or secondary metabolism is repressed. And in the production of these antibiotics, in order to actually maximize the production, you need to control the amount of glucose. And then we have non-ribosomal peptide synthesis. Some peptide antibiotics are produced independent of mRNA and ribosomes. So instead of the usual central dogma that we are doing, some antibiotics, they are produced via peptide. This is an enzyme called the peptide synthase. Example of that is gramicidin, echinocandin, and cyclosporin. And because it's an enzyme that produces it, it's of course under regulation. Strict catabolite or basically enzymatic regulation. So here is an example of your gramicidin. Okay, another one is autoregulation. This involved in a small molecules such as isocaprylol, hydroxymethyl butyrolactone. So this is in part of the production of streptomycin. So autoregulation, basically these are they usually function. This molecule functions as a hormone-like regulatory compounds. And another one is end-back regulation. This is feedback inhibition. In feedback inhibition, you have the repressor of that pathway is the end product of that pathway. So basically, this end product regulation tells that if you have produced enough of the product, you stop the production line. So here is chloramphenicol. Example is the production of the production pathway of chloramphenicol. So chloramphenicol, which is the ultimate product of this pathway, it inhibits the action of the first enzyme in the pathway, which is aryl amine synthase. And that's what you mean by end product regulation. The final product controls whether the pathway should proceed or not. And this is usually the way for the cells to tell that since we have already produced this amount of product, we should stop because we already have enough. So basically, that's your end product regulation. So here are some of these compounds. So inducible effects. So aside from metabolites in the same pathway that regulates them, we also have inducers from the primary metabolic pathway. So these are usually utilized in industrial production of some antibiotics. So example here is the production of, or rather the presence of methionine for cephalosporin production in your Acrimonium chrysogenum, and also tryptophan and analogs of tryptophan for ergoline in Clavirus purpurea. So basically, in the presence of these primary metabolites, they are primary metabolites because methionine and tryptophan, they are amino acids. And the presence of them induces the production of these secondary metabolic products. Aside from that, we also have the amount of nitrogen and phosphate can also have an effect on your secondary metabolism. For example, high concentrations of nitrogens or nitrogen sources or even phosphates can repress the production of antibiotics. So basically, when the product is a secondary metabolite, the condition must be strongly or rather strictly controlled because the excess of a certain chemical or a certain metabolite, even if it's a primary metabolite, can have an effect on the secondary metabolic pathway. Another one is the presence of ammonia that can repress penicillin production or even phosphates for candicillin, streptomycin, and tetracycline. So here are these examples. Okay, so that's it for the secondary metabolites or the lecture on secondary metabolism.